Several different dysfunctional immune responses have been associated with COVID-19, including the presence of anti-type 1 interferon autoantibodies. In order to broadly characterize autoantibodies in patients with COVID-19, Wang and colleagues used a new high-throughput multiplex detection technology they developed called rapid extracellular antigen profiling to discover that the patients with COVID-19 had a greater number of autoantibody reactivities compared with controls, and the highest scoring was in patients with severe disease. It appeared that some of the autoantibodies were pre-existing before infection, whereas others were newly acquired. Strikingly, autoantibodies specific for cytokines, chemokines, growth factors, complement components, and cell surface proteins were elevated in patients with severe disease, including anti-type 1 interferons. In vitro functional assays showed that some of these autoantibodies could inhibit the activity of the cytokines or chemokines that they bound, or enhance FcR-dependent phagocytosis, and autoantibodies specific for different blood leukocyte surface proteins were associated with decreased frequency of the cell types expressing those proteins. The scientists showed that blocking antibodies to several of the cytokine targets of the COVID-19-associated autoantibodies, including type 1 interferon, IL-18 receptor, IL-1 beta, IL-21, and GM-CSF, exacerbated disease. Other autoantibodies against various tissue antigens were associated with different clinical features and disease severity. This study demonstrates the power of a new autoantibody screening technology and expands on previous studies by showing that many different immunomodulatory autoantibodies are associated with severe COVID-19 disease. Further work will be needed to sort out the relative role of pre-existing versus post-infection induced autoantibodies in disease pathogenesis. Also of interest will be the analysis of autoantibodies associated with other viral infections.